Hello everyone, I would just like to welcome you today to Wattle's webinar titled More Than Just Dirt, Our Living Soil. So my name is Kim Blenkind and I'm the Executive Officer for Wattle, and I'm also a farmer in the Barossa Valley in South Australia. And I'd really like to thank all of you for taking the time out today to join us to be a part of this webinar. Today's session's been supported by the Murraylands and Riverlands Landscape Board through funding from the Australian Government's National Landcare Program and also from Landscape Board levies. And today we're really lucky to have Amanda delivering the session with us. Amanda's done some work with Wattle before. We've actually been out in the fields, but today, so we can reach a wider audience, we've decided to do this last session as a webinar. I'd now like to introduce Amanda really quickly, and then Amanda, if I miss anything, you can add just before you get started. So Amanda's a research scientist specialising in soils and land management with PIRSA. She enjoys delivering creative sessions that are packed full of things about soil, and she also loves talking about how do we make our soils healthy and productive. A little thing you might not know about Amanda is that she is also a netball coach and an avid supporter of her local netball club. And where we're not talking soils, we're talking netball. So thank you, Amanda, very much for joining us today. And I'll probably just hand over to you. But like we said to everyone, let's make today's session really, really interactive. Put your questions into the chat box or put up your hands if you have a question. But Amanda, over to you.
Hi everybody, thanks for joining us. It's lovely to do this as a webinar. As Kim was saying, we often do this in the field, and I love doing either venue. So I've been working in the soil area for just over 25 years. The first 10 years was working in soil surveys, setting up vineyards and orchards. And the last 15 years has been working more in the broadacre space, particularly in sandy soils. And my specialty is on soil carbon, so trying to get our soils to function better. So today is really relaxed, as Kim said, so I hope everybody's got a cuppa. Feel free to use this as a chat or a discussion group. So I will put up some ideas, and I'm really happy for you to stop and ask me questions as we go along. Otherwise, I do have sections there where there'll be a screen up and we'll just ask questions, because if you're like me, I forget my questions by the time I get to the end. So feel free to put your hands up and ask questions. So today we're going to be talking about exploring who and what makes a healthy soil, how we can measure our soil function in our agricultural systems, and how we're able to monitor this, particularly on farms, so things that we can do ourselves. Sorry, now my screen, there we go, it wasn't working. I can ask everybody why it's important to look after our soils, and everybody here will come up with a fantastic answer. So the key one that we're looking for is soil health. So soil health, or it used to be called soil quality, is not a new concept. We've been looking at this since the 1970s, but it's probably taking us a while to define what it actually is. So when we're thinking about soil health, it's this nebulous idea, but for us to be able to measure it, it's about how we measure how the soil's functioning in relation to that soil's natural capacity. And when we're talking about natural capacity, it really comes down to your soil type or your soil texture. So when we have a sandy soil, we know that it is inherently less fertile. Its capacity to be able to hold on to organic matter or protect it from decomposition is much lower than those that are more loamy or clay loamy soils. So we always have to think about our soil's function and our soil health in relation to the soil texture or the soil type that we have. For us to have a healthy soil, it needs to sustain biological productivity. It needs to maintain environmental quality. It really does need to promote plant, animal and human health. And this is probably the more relative concept in probably the last 10 years where we're really focusing on this. Soils also need to be resilient, profitable, and because we're talking about agricultural systems, they need to be productive. So we can't take productivity out of the equation. We need it to be all of these things. And that is a big ask. There is also no universal soil health benchmarks. We often have these things, can we have a single scorecard or can we use these metrics to measure soil health? It gets really difficult because one, our soil texture creates these differences about what their capacity or their inherent ability is to be able to provide it. And two, we all run different agricultural systems. We have different rainfalls, we have different setups and management that we do. So soil health benchmarks are going to be localized and can be used on your farm for the soil texture or at a regional scale. And we often hear about comparing our soils back to native or natural ecosystems or native vegetation. It's a pet peeve of mine because we ask entirely different things from our agricultural systems than we do from our native vegetation. Native vegetation is a closed loop. We don't take anything off of them. The biomass is there, so the biology is turning it over. With agricultural systems, we remove crop in yield, we remove biomass pasture for our livestock, we take off hay. So we are having these continual outputs, so they go off farm. So it's very unfair to compare these two systems. So if we are looking at soil health, we need to look at soil health of our agricultural systems. There are 5 key functions that we can measure that soils provide. And we have the first one is plant productivity, which we do really well. We can measure yield, we can measure biomass coming off the pasture, we can do it with remote sensing these days with NDVI or that fractional cover. And this remote sensing is probably going to be used more and more as we really get to understand that and ground truth it. The water story is incredibly important for Australia, but particularly South Australia. So that capacity of our soils to be able to, one, infiltrate, so capture all of the water that goes there, and then also hold on to the water that has come in. So that really comes down to the structure of our soils. So that soil structure and what we can do to improve it will help us with our water circulation. Nutrient cycling is probably the first thing that I hear that comes that people are thinking of when we're talking about function. We need to be able to get nutrients. And we do this by having organic matter in there, and we do this by having this amazing biology that decomposes the organic matter, which provides these nutrients to our soil, either from organic matter or from synthetic or chemical fertilizers. And so this nutrient cycling is incredibly important for us to have available nutrients for our plants to be able to grow. The key component of this, the next one, is the biological organisms. Without the biology in our soils, we can't have any of these functions. So organic matter and biology are probably two of the key things that we really need to understand to have healthy functioning soils. So the organic matter, the materials that we provide there, are the energy source for the biology. The more different varieties of organic matter that we can provide, either through above ground shoots or more often below ground roots and the exudates that occur in there, the more diversity of organisms we have to break down those organic matters. The more diversity we have, often the more disease suppressive these soils can also be. And of course, there's the function of greenhouse gas mitigation or sequestration of carbon in our soils. There's strong focus for the last 10 to 15 years on that, and that's probably one of the biggest areas that I work in. So it is an important component, but the difference in this function compared to the other 4 functions is when we're talking about greenhouse gas mitigation or sequestration, we really are talking about keeping the organic matter or keeping the carbon in the ground and not having so much of that decomposition. When we're talking about all the other functions, we really are relying on that organic matter to be turning over and decomposing. So we can get those benefits. So it all comes down to how much of that carbon dioxide is lost from the system. So when we talk about what is soil, I often hear the word dirt, and I think my daughter was about 7 before she started correcting everybody that it's not dirt, it's soil. But when it is dirt, it's when it's just those soil particles, when we're talking about gravel, sand, silt, and clay. So if we're just talking about those physical fractions within the soil, that does create dirt. When we add in organic matter, and that can be as a plant or animal, dead or living, and particularly when we're talking about this is the biological component, which then create, we have also air and water movement in there for our roots and for our biology. That creates soil. So we need to have the physical components, but we also need to have that organic matter in there to create soil. I don't need to tell anybody this, everybody knows it, and that the reason that everybody's coming to listen to Healthy Soil is that we know that soil is a precious resource. We have in the past had some events where we have lost significant amounts of soil, and nobody wants to go back to those places. So it's important to protect what we have. And when you're in some of those areas, so anybody in South Australia, those granite outcroppings, so if anybody knows bare rock around here, Tunkillo, it's made from granite. It can take 100 years to weather down that to that rock to create a centimetre of soil. So it's the reason why we are really conscious of protecting what we have. The biggest thing that you can do on farm, and this is what we normally do in our workshops, is really understanding our soil. So getting out in the paddock and being able to do a few things on farm to get a feel of what we have. When we know our soils, we're able to set up benchmarks or realistic criteria that we could see that our soils have the potential to reach. And undermining all of this really comes down to that soil texture. So the amount of sand, silt and clay in our systems. And in reality, it comes down to that clay fraction as probably being the most important, because one, it's the provision of nutrients. Two, it also has this ability to be able to hold on to nutrients, or particularly cations, and we call that cation exchange capacity. And three, it also determines how much water we're able to have. And when we're talking about carbon, it is the part that actually absorbs onto or holds onto the carbon and protects it from decomposition. It's called mineral associated carbon, and it stays in our soil for a very long time. So unfortunately, when we're talking about sandy soils, which I work in a lot, the benchmark or that capacity is lower for sands than it is for those loamy or clay loamy soil types. Soil colour is really important too. It can tell us so many things. The darker your soil, often the more organic matter you have in there. If you have those very bleached sands that you can see in that picture there, it often has very low organic matter, and you're thinking of really beads of little sand or beads of silica in there. Red soils often are a good aeration, and that bottom picture there with that funny looking mottling is an indication that those soils are wet for a long time. The depth of your soil layers is also really important, and particularly the depth of your topsoil. So where most of your roots grow, and that depth of your roots is a key part. So if you can take a shovel and have a dig around now that the soils are wet, and have a look where your roots grow, it can tell you so many things. It can identify any restricting layers that stop your roots from growing. And so from that, you can identify those constraints to plant production that may be there. One of the key things that I think is a significant investment is a field pH kit, which you can get from many of the hardware stores. Knowing our pH really allows us to, one, understand where our nutrients are available, particularly if they're at either end, so acidity or very in that purple end, that alkalinity. It can tie up or make nutrients unavailable or too available, so we can have toxicities. That's important for our nutrients and our plants, but there's also conditions where microbial biology is affected as well, particularly in that acidic end. If we can stop that microbial activity by creating conditions that aren't that effective. So knowing your soil really starts to set you up by having what that capacity is and identifying. And just remember, there isn't a bad soil. We have the soil type that we have. Our challenge, I suppose, is to manage what we have and to optimize that management to get the best out of what we need for our systems. Now, I just wanted to stop there to see if there were any questions before we move on to who lives beneath our feet. Was there anything there, Kim, or we're happy to move on?
There's a question from Jane. What is meant by environmental quality? So Jane, did you want to elaborate, come off mute and elaborate on that?
No, I didn't want to elaborate. It was on the first slide, and I thought, well, what exactly do you mean by that?
So the environmental quality part is often about, if you're near the Great Barrier Reef, it would be about the nitrogen or the leaching of nitrogen in there. If we're near waterways, it's about making sure that we aren't creating any environmental issues from our systems that we have. So healthy soil keeps everything as much as possible within the site that we have, rather than allowing it to move on and disrupting any other environmental area.
Right, so it's more related to environmental effects away from the actual site.
Yes.
Okay, so environmental effects might be
A better word.
Better word, yeah. Okay, thank you.
Any other questions?
No more in the chat box, but does anyone else have a question, want to come off mute at this point? No, I think you're right to continue,
Yep.
Amanda.
Awesome. So we're going to just take a bit of time and just talk about who lives beneath our feet. As I work a lot with school kids, so I go into schools and talk about soil a lot. And, you know, that their main thought about what happens with plants, or why soil is important, is to be that thing that holds the plants up. But we all know that soil is a living ecosystem. And within that, there are more people, there are more organisms in a teaspoon of soil than there are in the world. So it's quite amazing to get your head around that. So the things that we have here are the microbes. So they are the things that are too small to see with our eye. You can see it down a microscope. Bacteria is probably one of the most prominent ones. There's about 1 billion per teaspoon of soil, and there are thousands of species with so many different functions. But what we from an agricultural system like them for is that nutrient mobilization. And of course, there's also that plant associations for our legumes. We have the rhizobia, so without them, we wouldn't be able to fix nitrogen in those soils. We also have fungi, which, coming from sandy soils, they are one of those key components for sandy soils to be able to build structure. So they are the key component to binding soils together. And you get about 1 million fungi per teaspoon of soil. But what is really cool is you get about 200 meters of hyphae per teaspoon of soil. So that white, that webbing that we see is really important. They also recycle nutrients. And many of you will know that there are some symbiotic relationships with plants to access nutrients and what they need through mycorrhizal fungi. Then there's the protozoa. These are single-celled creatures. We get about 1,000 of these per teaspoon of soil, and they are the key recycling of nutrients by feeding on predominantly bacteria. And in doing that, they also regulate that bacterial community. So if you didn't have the protozoa, you can get the imbalance from bacteria to have good and bad species. And your nematodes, from an agricultural point of view or horticultural point of view, we often think of nematodes as something bad. We often measure them for those ones that attack crop or pasture roots, but there are many beneficial species. And it's interesting, in a lot of regen studies, the beneficial nematodes are one of those key indicator species that changes are occurring early. You get about 100 nematodes per teaspoon of soil. They are really good for recycling nutrients because they feed on everything else. So the bacteria, the fungi, and the protozoa, and those that do affect plant roots, they also feed on them. The macrofauna, so the things that we can do and that are easy to see with our eyes are one of those really good biological diversity measures that we can use on farm ourselves. And David Hardwick has created this rapid soil health assessment model, and that website up there is a link to download his book. And it has some really good guidelines, not just about the macrofauna, but about a lot of other tests that you can do very easily on site. And I've got that, we've got that in the references later. But he's basically divided them up into some 5 different categories. So you've got the things that are no legs, so they're the earthworms. We have things like 6 legs that are springtails, which have shown to be quite good indicators of soil health. Ants, dung beetles, termites, earwigs. 8 legs, we've got spiders and mites, and for anybody who is sensitive to spiders, I've chosen mite instead. And we've got things with many legs, so centipedes, millipedes, and slaters. So within these, what you would do is you'd take a 20 by 20 by 20 centimetre bit of soil, put it in a tray or on a piece of sheet, and you would assess how many of these different categories you have. So earthworms and dung beetles are really important for soil structural components within our soil, and add in ants as well, and they also affect the water infiltration. And many of these species will act as regulations for the pest and pathogen dynamics. So what we're looking for really is the more diverse number of species or these groups that we have, the more effective nutrient cycling that we will have, and that maintenance of pest and disease suppression and improvements in soil structure. So as a guide, when we're looking at it from the RASH model, if you have 1 to 2 of these types across there, they have that's moderate soil health. If you have any more than 5 of these, that's really quite good soil health. So how do we actually go along and measure soil health? And by looking at soil health, we're trying to measure changes in our soil function. So what makes a healthy functioning soil? We need to measure all of the 3 components. So from chemical, from physical, and from biological areas. The chemical area, we're really good at doing that. We've got pretty good standards. We understand that very well. We've been doing that for quite a long time, and we have a lot of soil test results that we could go back and assess that. The physical components, we're not bad at, but we don't often do it. So the physical components really is about that soil structure. And our biological components, even though we know it's probably the critical part, we still don't have enough information for us to be able to create some good standards for understanding how this relates to soil health. We know it's important, but we don't have those standards, nowhere near what we have for chemistry or physical. So in a soil, we're looking for chemical components to have a good cation exchange capacity, so that ability to hold on to nutrients and supply them, exchange them to the plant roots. We also need to have healthy pH, and the healthy pH is one for that accessibility of nutrients, but also creating an environment that's good for the biology itself. And low toxicities is what we need. When we're looking at the physical, we're looking for that structure and the stability of that structure, so that we're able to get good air and water infiltration and storage. And we need to ensure that the roots have good access to the nutrients and the water within. So not just in a shallow topsoil, we'd like to get them to get as deep as they can. For biological components, we need a good food supply, and we know that our organic inputs or organic matter is our key food supply for this. It would be good to have diverse and key functional populations present, and there are a lot of tests that are trying to understand that, but the more that we're doing DNA assessment and coming up with, we realise that we probably can only identify between 10 and 30% of all the biology that's present in the soil at this point in time. And the biological, the more that we have those diverse groups, we can have those disease suppression. And so these components within the soil help us then identify these soil functions that we have. And what I'll do is I'll just go through some ideas about each of these soil functions and what you might like to test in there or what's possible.
Amanda, there's just a question that's come in around the soil Urundi's test, the biological test.
Yes.
Do you want to talk about that now, or are you going to cover that a little bit later?
I will talk about that a little bit later, but I think it's one of the best tests. I love it. I think it's a great test to be able to do. So if you're happy, I will touch on that a little bit later.
Perfect.
So for productivity, for plant productivity or livestock productivity, we do this pretty well. You know, we measure this as a function of what we can do. So we want to, as much as possible, optimize production of plants to grow our food or our fibre off our farm. So biomass assessments often are in cropping areas. It's the yield that comes off. It can be dry matter, it can be those NDVI or that fractional cover. And as I said before, remote sensing is coming leaps and bounds, and we are getting much more confident in our assessments of this. So it's going to be something that we will be using definitely in the future, if not already now. For livestock assessment, probably the key one that is used is a kilograms of live weight per hectare. And sometimes when we're comparing across different regions, we're looking at it per 100 millimetres of rainfall, again, just to have that fairness about what can be grown due to rainfall. And there are also survival rates or genetics within that as well. When we're talking about our water story, there's bits and pieces that can go off to the lab, but the majority of this can be done on farm. So assessing your soil structure and how important it is for our water cycle is something that can be done yourself. So the aim is to improve the structure to get better infiltration. So we're capturing all the water that is falling on the place and then storing it. There are some really good visual and measures that we can have. So probably the key one of that is if we are digging using a shovel to have a look at our soil, we can have a look at the structure of the soil. So the size of these what we call aggregates, so what our soil breaks down into, and the arrangement of them gives us a pretty good idea of what the structure is. If we have a really heavy clod that's there, we know that it's not likely to have a lot of air or water movement in there. It's just too compacted. If we see that this picture here with these crumbles, so this is a light clay, those aggregates have broken down into these small parts which can allow air and water to move through freely. So that little crumbly structure is what we are trying to achieve. Just be aware that if you're in a sand, it is very difficult or it's impossible to probably get that. So again, about that realistic options about what your texture is and what you're expecting. Water infiltration is probably one of my favourite tests. We're finding it's quite sensitive for checking or for monitoring soil health over time. And I've got a slide that I'll show you in a minute. And soil strength and density is probably a key one as well. So this really picks up all of those limitations for root growth and for water movement in the soil as well. So soil strength is measured by a penetrometer, or you could use a piece of wire. And when the soil's moist, like it is now, you could go along and press down and find where it's quite hard to push. So you've got the easy ones, or you could put a 20-cent piece over a piece of welding wire or hard gauge fencing wire and push it down. If you get to a point where you can't push anymore with 20 cents on, you can't get it, that's the depth that you have a restriction. So water infiltration, the traditional way to do it is to get a measuring cylinder. This is just some storm drainage, so it's about 15 centimetres here. The traditional way of doing it is to put like an inch of water, so to make a mark on your inside, fill it up to 25 millimetres, measure the time from when you fill it to when it disappears, so there's just a glisten on the top of the soil. And from that, you can look at those millimetres, which would be 25, divide it by the minutes, and you'd get a rate of water movement into your soil. That can be very useful, and it gives you an idea of how many millimetres in an hour can come into your soil. But what we're finding much more effective, and Murraylands and Riverland Landscape Board has and Hills and Fluoro Board have a really good project where they've been monitoring the health of 5 sites with multiple measures in there for the last 5 years. And doing a little bit more with the water infiltration by, instead of just measuring that when it starts to when it ends, measuring that depth of water every 30 seconds for 7 minutes and then moving to that depth every minute thereafter and being able to plot it, you can get some really good data. And this data is one from a recent project in the Limestone Coast from the Grassland Society, where these lines here will give you the average rate. But what the important part is, can you see these steps that happen? So these steps are where water is getting held up. So we know there is a restriction of some type. Over time, what can happen if your soils are starting to improve is those steps get less and you get a much more steady infiltration of water in there. And that can be a really useful and effective indicator of if your soils are changing. Another good one that we've assessed, and again, this is this Grassland Society project in the Limestone Coast, where we were looking at comparing more stringent tests with some visual observations. So this was just looking at root depth, where we had a pasture soil and a vegetation soil. We looked at where the roots got down to, and we used a logging penetrometer to measure in 2 centimetre increments down the profile. And what we found was a really good correlation between what we were seeing for the rooting depth and where we were getting this key soil strength, or this root pressure that they can't go past. So 2,500 kilopascals is a pressure that roots can't penetrate. And so they're at these points here. So they correlate really well. So root depth is a very simple and effective way of being able to assess on site without any extra bits and pieces what is happening in your soils. Any questions to that point? We're still going okay, so we can stop and talk about it at the end too. So what everybody, and probably this is the key one that I get most people asking me about, is soil organisms and how we actually measure these. So we are aiming to get a better understanding and an improvement of diversity. So that's our key game, but how we actually get to this point can be difficult to know which test to use. So there are quite a wide range of tests. So visual assessment, such as doing that 20 by 20 by 20 centimetre shovel of soil and having a look at that macrofauna, is a really good indicator of what's happening in your soil, above your soil surface. The cotton strip test, which I love, which is a soil your undies, it's a really simple test, but it assesses that microbial activity. So for those who don't know, you can Google it, but what you're basically doing is getting 100% cotton material, you're putting it into the soil, and you can do it at different depths, but it's often the 0 to 10 or 0 to 15 centimetres. You bury your jocks or your vest or your piece of calico strip that you get from Spotlight. You put that into the soil and you leave it for 6 weeks. Now it's best done when there's a bit of moisture and a bit of warmth in there. So often late autumn or spring, probably mid-spring. Leave it in there for 6 weeks. Mark it, because when you go to dig it up, sometimes you can't find them. Pull it out and have a look. And again, the more activity you have, the less cotton that is left on there. So it's just a good visual indicator. There are some simple chemical proxy measures that you can use for organisms, and often this is just your organic carbon Walkley black that you get on your typical chemical test. The more carbon you have there, the more food source, so the more likely that you are to have microbial activity there. There's a pretty good relationship between that. But the only problem is you don't know. You can sometimes get really high carbon results, but you might not have microbial activity because the environment isn't right. And that is often when we either have toxicities there, we can have pH often very low, or we can have saline conditions, which can really affect the microbial activity. So we get this fake carbon value where you can't use that proxy measure to assess your microbial activity. And there are a number of specialised tests. So there are more and more these days. You can get some tests for respiration to understand that microbial activity. So that's basically a laboratory measure of your cotton strip test. You can have a test for abundance and diversity. So you can do that either looking down a microscope, or you can do it using a kind of like a DNA test for species and functional groups. We are getting more and more knowledge about microbiome and identifying which species go into which functional groups, but we are still probably lagging behind. So often a simple test is the fungi to bacterial ratio, or just a count of diversity within set functional groups like we were talking about before, the bacteria, the fungi, the protozoa. Now I will stop here because I'm sure there will be a question or two coming from that.
There's just been a bit of discussion, Amanda, in the chat box about the calico test, particularly from Alice, I think it was. Alice, and that landscape board might be doing a version 2, and it would be good to see it done across the whole state, was another comment. So I don't know whether anyone wants to comment on that.
So I'm Alice. I do citizen science at the landscape board. We're just in talks about doing a version 2 at the moment. We haven't fully decided on what's happening, but I can definitely be in touch with the people here when we actually have decided. And we can talk to other boards about what they want to do as well. It's a fairly simple thing and cheap to run as well. So hopefully they want to get involved.
Excellent, thanks, Alice. And are you with the Murraylands and Riverland board, Alice?
Yes,
excellent.
I am.
Excellent.
Yes, sorry, just to add a bit of context to that, just in case people didn't know, we ran a Soil Your Undies citizen science project, I'd be 2 years ago now, Alice.
Yeah.
So yeah, it'd be version 2 of that, and that was nationwide. So that was done through Southern Cross University, and actually caused a national Y-front jock shortage.
Oh dear, we don't want to do that.
So many jocks were bought, so many grumpy grandpas. So yeah, we did do that, and it was a run in spring, and we sent out over 200 packs of jocks in the region, and other organisations did that too. So at that stage, the other boards weren't involved, but yeah, as Alice said, we're certainly happy to get something statewide going, and yeah, it is relatively cheap to run. The only real cost is the purchase of the jocks. So yeah, that's just a bit of extra context there.
And so what happened to the information, or Alice, what happened to the information that was generated from that?
They would send their jocks back to us, and then we would dry them and weigh them. So that was quite a lengthy process. And then a small summary report was provided back to the participants about their jocks. So what happened is they did paired sites. So we encouraged them to pick somewhere like a driveway or a path, something like that, where there's, low visual organic matter, high levels of compaction, and then compare that to something that, whether it be a compost heap, a veggie garden, that type of area, just to see how active some soils are and how active and inactive some soils aren't. And it was then buried on the same day. Like Amanda said, mark where you've put them, because there was lost jocks. But some were, you could pull them out of the ground and wash them and wear them the next day, and others, all that was left was a piece of elastic. So it was quite a engaging way and simple way to see how alive your soils are. So yeah, there was a report provided, and we still do have photos and information on that first phase 1 of that project as well, if anyone's interested.
And I think the important part, like that, you said, it shows those distinct differences. It's a lot harder to use if you're looking for within a system that's quite functioning, to be able to measure those little changes. So I think once you've got past that and you want to do it, then you go on to another one of the tests. But I think it's one of the most useful tests for getting people to talk about soil health and microbial activity that we have. Okay, I will continue going.
Oh, Amanda, there was just a comment
sorry.
from Karen in the chat box. Karen, did you want to elaborate on your comment or ask Amanda a question?
Yeah, I suppose looking at that next slide, we might cover it. It's just about the relationship between soil moisture and microorganisms. Yeah, if we can have a look at that, but we might be heading there anyway.
Yep, and that's a really good point, because without moisture, most biological organisms will go into not hibernation, but they just go into stasis, so that they can't do anything. They need to, same as us, have water and food and temperatures available to be, or temperatures in a good range to be able to be active. So we often find that's why we do that late-ish autumn with moisture, or that early spring when we're actually taking biological samples to focus on those. There's no use taking it when it's really wet in winter and it's really cold, or it's really hot and really dry in summer. So that's a really good point, Karen. And for us to measure the function of nutrient cycling, this is where we have fantastic standards. We've got, we know about plant tissue, probably not so much about SAP tests, but SAP tests are being used more and more often. So as we get more information, standards will probably be created. Soil tests, we have some really good ones. So we have good standards. And really, for the understanding of soil health, it often comes down to that pH, so being in that range where nutrients are available, but that also is conducive to biological activity too. Our soil carbon, that organic carbon Walkley black, is important when we're talking about soil function. And we also have cation exchange capacity, and I like to use the cation exchange capacity as that measure of the inherent capacity of your soils. So often when we're talking about sands, we're talking about cation exchange capacity between 1 and 4. When we are talking about things like a loam, we could be up around that 15 to 20 range. So there's a lot of difference between that and just that ability of what that soil can do. And the last one, the function that we talk about is greenhouse gas mitigation. So this is where I spent a bit of time trying to understand our potential or our opportunity in our South Australian soils and how we could possibly increase these. So when we talk about greenhouse gas mitigation, we are talking about taking carbon out of the atmosphere and storing in our soils or sequestering in our soils for a long time. So 25 to 100 years. So that's a long time. And I always like to have that proviso to increase carbon where possible. Because again, as I said before, for the other 4 functions, we are really looking at using that turnover of that breakdown of that organic material to release the nutrients and for improving our biological. As we do that, we have decomposition and we have carbon dioxide loss to the atmosphere. So soil organic carbon tests are important. The one that we often test is organic carbon Walkley black. That comes with all of our tests. That's great for looking at those other functions. But when we're talking about greenhouse gas mitigation, we need to talk about total carbons or total organic carbons if we have calcareous soils. And this presentation will be available as a PDF, and I have a range of tables at the end which will go through all of the soil organic or soil carbon tests and tell you which one's the best for doing whichever monitoring. So for these, the crux of this one is that we often are expecting carbon to have a dual purpose in our soils. So for function, we wanted to improve it. We need to decompose it for nutrient cycling. For greenhouse gas mitigation, we want to store it by protecting it from decomposition, or we want to transform it from the active to stable fraction. So again, biology is the critical component in doing these, and it can often be that scales about which way we're tending. So management activities can change depending on if we want it to be cycling or we want it to be storing. So if we go through the difference about carbon, where it comes from. So plant residues, the majority of the thinking probably 10, 15 years ago was that the majority of carbon that was in our soil comes from above ground. But although the above ground component is incredibly important, it is there, it creates the environment, moisture, temperature protection above ground. It's the below ground, so it's really is those growing roots and those root exudates that create most of the carbon that we see stored in our soils that is there for the long term. Fascinating, it's the soil biology that creates quite a bit of the carbon that we measure. So the living biology can make up 1 to 5% of the carbon that we measure, and the dead microbes or the microbial necromass makes up 30 to 50%. So this is where we see how important that living biology and particularly the bacteria are for changing our carbon. So it's that trade-off between having that cycling and that building of microbes and then having it still keeping there in a stable form in our soil. And we also have inputs from manure and urine. We can have compost and manures coming from off-site. And we have what's called a long-term stable or recalcitrant sources of carbon from fire or biochar. So what we remember is that what we measure in our soil is that balance between what we put in and what we lose. And decomposition losses can be between 70 to 90% of any carbon that gets into that soil. So we need to think of soil as a sink, so that storage of carbon, but it is also a source from this decomposition of greenhouse gas emissions. So we need to get that balance, and that's what we're trying to do in lots of situations is optimize that balance so that we can have a functioning soil, but we are still improving our carbon. I could talk to you all day about soil carbon, so I've only put in a couple of slides, but probably the key takeaway factors are that carbon, there are potential upper limits that are defined by your soil, and it comes down to, again, that texture. And in this component, it's the clay, because it's the clay that adheres, the organic matter or the carbon that adheres to the clay that is there for the long term, that stable component. Limiting factors such as carbon then define how much organic matter inputs can be grown, and in South Australia it really is that rainfall and temperature component. And then we have our management decisions that we make. So that's the next step down that we have, and it really does come down to tillage and the ground cover. So the amount and the length of time that we have ground cover or actively growing plants has a big impact on our carbon. The biggest question I get asked from any farmer is, how do I know if my soils can store more carbon? So we worked with the Department of Environment and Water to analyse a heap of soil tests that we had when we used to be a state lab. So we had 36,000 soil test results during the time of 1990 to 2007. They were from the 0 to 10 centimetres. They were using that test, that organic carbon Walkley Black, which is a standard test that we get when we send it off to the lab. And we were able to have a look at trends over time, and we could have a look at different components. But probably the most useful thing for people and farmers that I've found is these benchmarks that we've created. So remembering these benchmarks are based on 1990 to 2007. So we have no idea what's happened from 2007 onwards. We don't have that data anymore. But for each of our agricultural districts in South Australia, we have these tables, and the information that you get given. So that green box tells you that 54% of the samples were from cropping, 31% was from pasture. So this is the upper southeast. The trend, they get an annual trend of what the carbon occurred over that time. So this was a 0.01% trend, and it was relatively stable. So it was up and down. So it evened itself out. But the key thing is that if you have a 0 to 10 centimetre soil test result that you've got either in the filing cabinet or you've got some new test results this time, you get your 0 to 10 centimetre sample and you compare it to what your soil texture is. So you may fall in between at the higher end of the limit. If we're sitting at these higher end of the benchmarks, we don't have really good guidelines. So these are all my gut feels. So you can, if we're sitting at about 60%, the opportunity to increase carbon is probably much harder. So you can still do it for sure, but the opportunity or the increase will be smaller. If you sit below 25%, I would say there is some major constraint, soil constraint that's occurring in your soil that is affecting how much carbon can be there. And you would need to assess if that is practically or economically feasible to change it. If you're anywhere between this 25 to 60% here, there is that opportunity that you could store more carbon in your soil. So you have to really use these as that idea about, where am I sitting at? What's my starting level? Can I achieve that 75th benchmark? And can I maintain those inputs once I get there? So it comes down to these management options that we have. So we want to either optimize our carbon inputs, or we want to minimize those carbon losses. So wherever possible, with your root depth that you can look at on your place, you can address the soil limitations. So one, identify them, then work out if it's economically or practically feasible to change it. And in some situations, it's not. Where possible, you can optimize nutrition. The more where you have that nutrition factor, we can, one, grow more biomass, but two, we can also aid that transformation from that active, that quick turnover fraction, to that more stable fraction. The idea of growing green plants for longer and having more plant diversity and swapping out with perennial instead of annual plants is being really widely adopted and very easily done so in pasture systems. In cropping systems, it is much harder to do, but having said that, there are some fantastic examples of people where they're either companion cropping or in areas where we have enough rainfall, being able to summer cover crop over time, just to try and get that green for longer. So there is the option of providing organic matter inputs, and that's often from externally off-site. But having said that, if you have a piggery or a dairy or a piggery chicken close by, it may be worthwhile looking at that, and there's quite a few people who are currently doing that. And optimizing carbon inputs comes down to minimising bare ground, which also flips over to that minimising carbon. So that is that's through decomposition of carbon, but it's also from erosion. So minimising soil disturbance and bare ground is probably your key parts for minimising erosion. And again, that capture and stabilisation of carbon is that decomposition story where we're trying to take it down from that 90% of carbon being decomposed and lost to the atmosphere and bringing it back to that 70% part. And that's where there's quite a lot of work being done at this time to get those activities that can do that. So our soils, they are living, they are an incredible ecosystem that we need to look after. So we need to look after it from the physical components of the point of view, but from the fact that it's a biological entity that we also need to feed and look after as much as anything else. Organic matter is your key component, it's your energy source, and it's also very important for those functional benefits when carbon is decomposed by the biology. There are a wide variety of organisms within our soils. They are adapted to different soil conditions. So it's that different soil texture, it's those different chemical ranges, or it's that different pH. But we also have to be mindful that we need to create soil environments that as much as possible supports the most biological activity that we can have. If we are focusing on that soil health and having those functional opportunities to improve our water, for our nutrients, for our plant productivity, it's really important that we do have those environments. And it's really important also, if you are thinking about it from a sequestration point of view, that we are optimizing those microbial efficiencies. And I think that's going to be the next exciting thing for the next 2 to 5 years where we come out of. This excellent slide from How to Build Soil Health from Graham Sterling. There's a book, and I've got the reference in the next page, about what you can do to build your own soil health. So it goes down to assessing those physical, chemical and biological components of the soil that we've spoken about, identifying those constraints, identify if it's feasible to change it, and then look at those options for managing it. And the key part is then monitoring this. So when we're talking soils, nothing happens quickly, okay? So we are talking about monitoring in the long term, and it's often 3 to 5 years before we see some decent changes, and it can be often up to 10 years before we see significant change, okay? Particularly when we're talking about changing over soil structure, it can take a long time. So photos, if you're going to do it on farm, photos are your best friend, because no matter what, you won't remember when you go back to your site what it was. And in a site, we know that landscapes change quickly. If you're in the Mallee, you've got dunes, you've got swales, so you've got sands, and you've got a loamy or a clay loamy soil down there. Pick an area that's a paired patch, like Zoe was saying before. Have those paired comparisons to give you an idea on farm or in paddocks to see what is changing. For soil health, we are building carbon as your food for your microbes. So we're all about feeding the bugs in this one. For sequestration, we are looking at protecting carbon from decomposition by microbes or increasing that efficiency. So there are slightly different purposes about what we are doing. We're not going to go through these, but these are examples about what will be in the PDF if you're available. So if you wanted to assess soil function, that's got your interest there, and then it's got an idea of how you can measure it. And then for each of those ones, I've gone through what tests you might use, what the methods are, and the pros and cons, okay? So it just gives you a little bit more information to follow up on. And there's some reference sources too, just for South Australian context, and there's some good books like the Soil Health book, and the detailed info on biology tests is in there, and David Hardwick's rapid assessment of soil health. It's a really easy to use on-farm system that I really like. And from that, I think I'll leave that one up there for Kim to talk about afterwards, but we'll just check if there's any other questions.
There is a couple, Amanda, and it was back when we were talking about the chemical stuff around soil, but I didn't want to interrupt you, but there's been some discussion in the chat box around testing for soil phosphorus, and especially on calcareous soils, and the use of the Coal Whirl P test and potentially the PBI or the So you know these acronyms off the top of your head. I'm going to read it. The DG, yeah. So can you just have it, you know, so I suppose the conversation's been around what's the best way to test for phosphorus in calcareous soils. So do you want to talk a little bit about that, Amanda?
Sure is. So in calcareous soils, we use that DGTP. So we get Colwell P on all of our tests. But for calcareous soils, we will go with the readings from the DGTP. It is shown over and over to be the most consistent test for calcareous soils. And in, I'll just flick up. There is Understanding Soils of South Australia. Within that, there are some ranges that include that DGT. So what you are looking for ranges within that for what would be acceptable below average in there as well.
Excellent. Thanks, Amanda. There was some discussion too around, I'm just going to scroll it, from Louisa around a carbon market workshop or conference coming up. Louisa, are you still online, and did you want to mention anything?
Sure. Hi, Louisa Kiley, Carbon Farmers of Australia. So we've done 10 carbon farming conferences and expos, and this year we're calling it the Nature-Based Solutions Conference. That's because there is also another market coming, which is this biodiversity market. And so anyone who's thinking about improving their soil health, which improves the health of their country, biodiversity above ground, biodiversity below ground, grazing situations, all of those, there is potential now that we can also engage with what's called the nature repair market, and that's only just starting. And so we're talking about that at the conference, but we've also got a full day on soil carbon, and we're going to showcase. The government had the Soil Carbon Challenge just prior to the election last year, so a year or so ago now, and the previous government put in, I don't know, large amounts of money for people to, the deal was reduce the cost of measuring your soil carbon and your soil health, if, you know, with some of the other things thrown in there. And so we're bringing the update on that. Tower fluxes, all of these done under this Soil Carbon Challenge, Innovation Challenge. We'll be talking, a farmer will talk about his experiences in a soil carbon project. It's just a way to sort of start to think about, if I'm thinking about soil health, soil biology, and the relationship to soil carbon, do I want to, you know, engage? It's what we set out to do is to give the education around how this sort of fabulous information can lead to a market if that's what people want to do. And we sort of pride ourselves on the plain English grassroots version. This isn't at a corporate level. This is at the grassroots level that we explain how it works. And then we go on to the other days and do nature repair markets and supply and demand, and where might the price of carbon go, all of those.
Fantastic. Thanks, Louisa. And the email address, or an email address, a website address is in the chat box, and I will pull that out and send it out to people interested in following up on more of that after the Zoom. So thanks, Louisa. Is there any other quick questions? We're actually past 11:30, but is there any other quick questions for Amanda? I think we're good to go, Amanda. So can I get you to flip through to the next slide? Yeah, so just some Wattle events that are coming up. That's our address to get to see all of Wattle's events. But our big ones is that we've got our Regenerate Rural Women's program starting on the Yorke Peninsula, Murray Bridge, Tintinara and Pinaroo in the next month. So that's been a really successful program for rural women. And also we've got our Thriving Women's Conference in Wagga in August later this year. So again, if you go onto our website and have a look at those. And then if you flip through to the next slide, sorry, Amanda, to make you do this for me. Today's been has been funded with the support of the Murray Lands and Riverlands Landscape board through funding from the Australian Government's National Land Care Program. So as a part of that funding, we need to do some evaluation. So I will send this QR code out to everyone as well, but you can actually do the evaluation now, and I'd really appreciate it. So it's just a matter of holding your phone over that QR code, and that will bring up, and so Zoe's doing it now, that will bring up an evaluation form. The event you need to scroll down to and pick is other. And it just takes a couple of minutes to do the quick evaluation on your phone. So I would really appreciate it if people could do that for us as well. Amanda, a big thank you to you. Again, you make what I think is a quite technical and scientific topic really, really simple for people to understand. So thank you for sharing all your knowledge and expertise in such a great way that we can absorb it and go out into the paddock and actually do something to measure our soils and what we're doing. So thank you again very much for today. I'm going to turn off the recording now.